Okay, so in this video, let's talk about the synthesis of the MAPP and IPP. So this is not that much complicated one, but there are some few steps you need to be very careful of. Just a simple reactions. So hang along with me. First of all, we are going to start with this molecule. So I have drawn two molecules, right? So in these molecules, for this carbon there are hydrogens. These are alpha carbons, which means this hydrogen contains this hydrogen have relatively higher acidity. So with a proper biological base, you can remove this hydrogen and create a lone pair. So let me draw the lone pair here. Here we have the lone pair. So this lone pair can act as a nucleophile, and this is a carbonyl group. This carbon over here has an electrophilic characteristic, so nucleophilic attack can be happen here. So in this first step, a Claisen type reaction happens. So if you know about the Claisen type reaction, I hope you know, because the Claisen reactions are very basics of organic chemistry and both biochemistry. If you have not remember it, or if you have no idea about the Claisen reaction, I highly recommend you to go back to our previous videos and watch the Claisen reaction mechanism. So what happens actually is, when this nucleophile or this lone pair attacks to this carbonyl compound, this bond breaks, right? So again we have now a double bond here and negative charge here. Now in the Claisen mechanism, what happens is, right, in Claisen mechanism, this lone pair forms back again to form the pi bond, and here we have our final product, right? Here we have the final product. So if you remember, in the aldol reactions, when this bond is broken, right, it won't restore the pi bond again. It will go and attack to a proton and form a hydroxyl group. In Claisen mechanism, it will form again the carbonyl group. So here we have the first type reaction, and then we have another molecule here. So here the same molecule. Again the Claisen type reaction happens, right? This way, this Claisen type reaction happen. Sorry, not Claisen type reaction. Now the aldol type reaction happen. Aldol type reaction here. So with the aldol type reaction, you can see here, this will go and attack to this place. Now aldol type reaction, when this bond is broken, right, it will not restore the pi bond, it will go and attack to a proton, right? So now look at very carefully, right? So here we have the carbon, right? So this carbon, this is the carbonate here, okay? This is the carbonate here. So this is the carbonate here. You can see this carbon, I will mention it as number 1. This is over here. This is number 1 carbon. So this bond is the bond between this carbon and this carbon. Now you can see this oxygen over here. This is the place where the aldol type reaction happens. So now it has converted into a hydroxyl group. Now this aldol type reaction followed by the basic hydrolysis, which convert this enzyme part into a carboxyl group. Now we have this molecule over here. I hope you understand up to now. It's very important to understand what's happened, right? Just do not memorize this mechanism, just understand what happens. These are basic reaction, basic Claisen and aldol type reactions. These are very basics. The only thing you need to understand is the picture, like how the molecules come together, right? So I hope you are okay up to this point. Now in the next case, we are using NADPH, right? NADPH to convert this particular carbonyl group into a hydroxyl group. Here you can see, now we have converted this carbonyl group into a hydroxyl group. So I have just put this whole part like this, the same part, right? There's nothing change over here. Only change is this carbonyl group change into hydroxyl group. And now in the next case, right, in next case what happened is, we're going to remove this coenzyme group and convert our hydroxyl group into a carbonyl group again. Okay, so here we have carbonyl group back again in your molecule. Now we have our NADPH. Now here we have a hydrogen, right? This is hydrogen. So this is our last carbon on the chain. So when we are adding the NADPH, this converts into a hydroxyl group. So we have this molecule over here, right? So I hope now you are okay. In the next step, so I have just drawn the same molecule, okay? Just the same molecule over here. We call this molecule as malonic acid. Malonic acid. If you are familiar with biochemistry, you are surely heard of this molecule. This is very important molecule in biosynthesis pathway, especially in malonic acid pathway. So now in the next step, what happens is we are going to use two ATP molecules. I hope you know what the function of ATP, right? ATP is in equilibrium with ADP, right? These are triphosphates, these are diphosphates. So with this one, this release a phosphate. So what happens is these ATPs, right? It releases a phosphate group. So normally in biosynthesis, we denote phosphate groups as P. So we are using two ATP molecules which contributes two phosphate groups. So these two phosphates group replace this hydrogen over here. Here you can see two phosphates groups have attached to our malonic acid. Now this is our next step. And then what happens is a decarboxylation. So here we have the decarboxylation. And decarboxylation, you know, like here you have a hydroxyl group and here you also have a hydrogen group. So this decarboxylation and this double bond formation is also happening, just a basic elimination process. So which makes this IPP molecule, right? Here we have the IPP molecule. These two are resonance structures of each other, right? These two are resonance structures of each other. So you can get both of these two with biological acids and bases. So this is the basic synthesis of DMPP and IPP. These are very important molecules in our mechanisms and biosynthesis pathways. I hope you get the idea about it. So again, these are not complicated mechanisms. Just understand the picture, understand how molecules work inside our mechanism. That's the most important thing.